Okay, guys, let's talk about enzymes. So, enzymes. Oh no. There you go. We'll figure this out together. Let's do that guy. All right. So we have enzymes. Okay. Now, enzymes are a special class of proteins. Remember, we've talked about proteins before. Remember, proteins are made of the monomer amino acids, right? And remember, proteins, as far as function go, that's going to be they build the physical organism. All right. So enzymes, these are going to be found in living things. And it's actually really cool what they do. Another name for them, just to get it out of the way, is a biological catalyst, right? And a catalyst is just something that you would find in chemistry to make a reaction happen. And so enzymes, what they do is they allow, I'm going to put reaction like that, they allow reactions to happen in living organisms. And you guys realize that you have a bunch of different reactions happening inside your body all the time. Like if you guys eat sugar, right? So like let's say here's a sugar molecule and it's attached to another sugar molecule. When it comes inside your body, what it does is it breaks it down into smaller pieces, right? Because we talked about every time you break a chemical bond, when you break a chemical bond, then you get energy. So when you guys eat something and then you start to digest it, right? You break those chemical bonds. And when you break those chemical bonds, that's going to allow you to have energy. And that's why you get energy when you guys eat food. But this right here, the actual breaking of that chemical bond, that's because we have enzymes in our body, right? Okay, a few little things. First of all, enzymes always end in the word "ase," and "ase" is just a suffix. It's what it ends in. And usually the stuff that the enzyme is doing the reaction to is named for that. So like if we talk about lactose, remember lactose we know is a sugar because it ends in ose. The enzyme that breaks it apart is lactase, and that ends in ase. Or if maybe we have the sugar maltose, then the enzyme is going to be maltase. And so that's just really kind of how it goes. Now, enzymes, if we want to give them a nice definition, is they speed up a chemical reaction by lowering the activation energy, which is a really big, long, complicated definition, but it's pretty easy to understand. They speed up, so they make it happen faster, chemical reactions, by lowering the amount of energy they need. That's activation energy, that something needs to actually have it happen. And so if I look at two situations here, let's draw a real pretty picture. Okay, this is going to be hill A, and then this is going to be hill B. All right. So let's draw this little guy over here. So I have this dude, and he has a ball, and he has to roll the ball up the hill. Okay, so here's the dude, there's his ball. And so his job is to roll the ball up the hill, okay? And if you're looking at A or B, which one is going to be easier to roll the ball up? It's going to be A. Well, why do you think it's A? It's A because this hill right here, say he doesn't have as far to push it up. But B, this is a really big hill, okay? So this one would take less energy versus hill B, this would take more energy. And so really, we can use enzymes to look at this. Because if I look at hill B, this would be a chemical reaction without an enzyme. So could I push this ball up the hill? Sure, I could push the ball up the hill, but it's going to take a lot of energy. It's going to take just a lot of effort. But if I choose to push the ball up hill A over here, that takes a lot less energy, which is what enzymes do. Enzymes speed up the reaction by lowering the energy. And so this over here, hill A, I get to do a lot less energy, right? And so if you have a lot less energy, you're going to be able to make your reaction happen faster. That's why if we look up here, I speed up the chemical reaction by lowering the activation energy. So if I have this guy with the ball up the hill, he's going to be able to do this a lot faster with this hill because it's a lot shorter distance. So he can speed it up because the hill is lower. This guy over here at hill B, say it's going to take him a longer amount of time and it's going to take him more energy. And that's all enzymes are doing inside a living body system, is allowing reactions to happen more quickly. And we know we need chemical reactions to happen quickly inside our body because our bodies are living systems. And, like I said, think about digestion, think about respiration, think about any body process is probably fueled by an enzyme. And if it's fueled by an enzyme, then we need it to happen more quickly. Does that mean that these reactions will never happen? No, they'll still happen, but they may not be in a time frame that we need it to be in. So enzymes speed up the reaction by lowering the amount of energy. All right, now things that can affect an enzyme. Affect an enzyme. All right, since an enzyme is a protein and proteins come from living systems, I usually talk about puppies, right? Say we're all super familiar with puppies here. And so if you have a puppy in a living system, right, they can do all these really fun things, but when your system isn't in ideal conditions, then that could hurt your enzyme. All right, so effect of an enzyme, I don't like that pen at all. Let's do this pen. So effective on an enzyme, this could be like temperature, right? Because if you stick a puppy in an oven where it gets really, really hot, that's going to break the puppy, right? So if you stick an enzyme in a location where it's really, really warm, that could break it. Or if you stick your puppy in a freezer, that could break your puppy because it gets too cold. It's the same thing with an enzyme. So enzymes, they work at an ideal temperature. And if you go above or below it, so if you go above it or if you go below it, that could break your enzyme. It's the same thing with pH. Remember pH when you talk about acids and bases? So if you stick your puppy in an acid, guess what? That's going to break your puppy. If you stick your puppy in something basic like bleach, that's going to break your puppy because enzymes work in ideal pH conditions. So if you go above or below those different pHs, then that could potentially break your enzyme. And if you break your enzyme, then it's not going to function, okay? When we break an enzyme, there's actually a fancy word for it. So if you break an enzyme, this is called denature. Because D means not or undo, and then nature is your living thing. So I'm unliving something. That's when I break my enzyme. I unlive it. And I can unlive it by messing with the pH, by messing with the temperature. All right, let's end this by drawing an enzyme, okay? So let's make it really easy. Do enzymes always look like that? No, but they are today. So here's my enzyme, which is beautiful. And then I need the thing that the enzyme is going to do the reaction to, okay? And if I look, this looks like a triangle. This is called the substrate. The substrate, this is what I need the reaction to happen to, okay? Now, if you look at this, so I have that substrate, and let's maybe draw this substrate. So this could be a substrate too. And if I look at the enzyme, which one of these is going to fit inside the enzyme? It's going to be this triangle piece, right? So enzymes are super duper specific, which means they only fit, let's write words, say they can only fit a specific substrate. Okay, and this entire idea, which actually isn't too hard to understand, this is called the lock and key model. Because only one substrate is going to fit, the substrate would be your key, only one substrate is going to fit into your lock, which is your enzyme. Because if I do like a silly example, we all have front doors. And so if I take my key to my front door, that key is going to fit inside that lock. And then the reaction will happen. My door will open. But if I take my key, maybe my key's a triangle, and I go to this enzyme that has kind of a square thing, then that's not going to fit. So I can't take my front door key and go to your house and open up your front door because it doesn't fit, okay? That's where lock and key model come in, that only one substrate is going to fit into that enzyme. And so how these reactions happen is pretty neat. So here's my enzyme, and then here's my substrate. Remember, so only one substrate is going to be able to fit in this sucker. All right, so now let's do a reaction. So I have those two sitting there. Here's my enzyme. I put an E on it. And then my substrate is going to come fit inside that. This right here, where it fits, this is called my active site. So active site is where the substrate fits. All right, so now my active site, substrate has come to fit in. And so maybe this enzyme is going to break the substrate apart. So now maybe it looks like this. So I still have an enzyme, but now these, instead of being my substrates, these are my products. All right. And I can do the same thing. So maybe that was, because see, enzymes, sometimes they can, that's supposed to be the word break. Say sometimes these can break things apart. But other times I can build things. So maybe here's my enzyme, there's my E, and then there's my substrate, S. And so if I want the reaction to happen, there it is. And then this guy is going to sit right inside here because that's my active site. And this is called the enzyme substrate complex. And it's the same thing up here. Say when that enzyme sat inside the active site, this was my enzyme substrate complex. So I had right here, here was my enzyme. Here is my substrate. They come to sit together to make my enzyme substrate complex. Oh no, I drew the wrong picture, guys. All right, well, let's pretend up here this substrate. Let's pretend that substrate was two different things, right? So instead of being one big blue box, oops, let's undo that. We have two blue boxes. Those are my substrates now. And those go and sit inside there. And then when the reaction's done, I have one big blue box, and that would still be my product. And then if you notice on every picture, right, I still have that enzyme. See, there's the enzyme right up here. Here's the enzyme right here. So something to note is that enzymes are reusable.